Good afternoon, everyone. Hope you guys are doing well. It's a nice day outside, so I hope you guys got to at least enjoy some outside time. It's always good. I feel like college doesn't encourage outside time enough. I digress. So this week we are going to be diving into DNA. So we have we have our lecture like usual, and then we have a pretty cool. At least I think it's pretty cool. Um, but you guys know by now I'm pretty nerdy, so the things I think are cool probably aren't the things that you think are cool. But we're going to be doing a little exercise as we tend to do on a really kind of a fascinating and very important biotechnology tool called CRISPR-Cas9. And so I hope you guys all had a chance to read the read the materials prior to coming, because CRISPR can be a lot to take in for your first time. So in terms of stuff, you have a homework that's due this week. There's your textbook chapter. Your annotated bibliography for your BioWiki is due this week. So, you should be reading sources. You should be summarizing the sources and submitting that. Just as a reminder, it's due Friday by midnight. And basically, this is what you need. It's pretty straightforward. You're just reading and annotating. And I have you do this not just as a um, I guess I could say torture. It's not just busy work. This actually is a really good way to just get you to write down a lot along the way. So annotating your bibliography is a great way to sort of know what you're going to say based upon what you read. And so basically, when you do this bibliography, you basically take all this stuff and you basically stick it in your paper. So it is basically a way to get you writing your paper early on and thinking about your paper early on. So, um, you know, it obviously is part of your grade. It's 10 points, so it's 2.5% of your grade. So it's not a huge percentage of your grade, but it certainly pushes you in the right direction, thinking about your wiki, because you don't want to leave this till the end of the semester, and that's just not going to help you out. So, and again, it's nothing crazy. If you're spending hours and hours on this, you're working too hard. So, if you're having difficulty with the bibliography, please let me know. It's pretty, like I said, it's not super in depth, but again, it gets you thinking about your topic, gets you putting sources together, gets you, doing all this stuff. So definitely want to do it. And obviously, if you have any questions, do let me know. And the other thing is, before we get started, you guys did pretty well on your first exam. In general, there's always outliers and people who exceed or don't make it to sort of the class average. But the class average was pretty good. It was around like a 32, I believe, which is it's in the 80s, so that's pretty good. So you guys have a general flavor of what what the exams look like, what the sort of flow is, and sort of the questions I'm going to ask. At some point I'll send out, before the next exam, I'll send out like a little survey to to get your feedback on the first exam, see what you think. It'll be anonymous, obviously, but get what you think. Is it too hard? No one's ever going to tell me it's too easy, because people don't do that. But if it's too hard, the material's too abstract, it's not close enough to lectures, any number of things. So I'll send out a survey to see what you guys think. But again, you guys all did pretty well. So, I think, it won't get harder than it was. Let's put it that way. It only could potentially get easier. So. But again, if you have any questions about any of the things on your exam, you should be able to go back and view the exam, see which questions you got right, which ones you got wrong. If you have any question as to why you got was wrong, or you want to argue with me about, about a question, and I say argue not as in like angry arguing, as like just discuss your question. Obviously, we're not gonna argue with one another. But if you have any questions, just shoot me an email and let me know. So, all right. So that's pretty much it. You did have no lab due this week because of Columbus Day, just as an FYI. In terms of Labster stuff, if you can keep doing Labster. Like, you don't have to do it the week assigned. You could do all the Labsters in one sitting if you so chose, just as an FYI. I see people are doing them 1 week at a time, and that's cool. But like, if you wanted to be super on top of things, you could do all the Labster things in an afternoon, because the simulations are only like 40 minutes or so. So So we will have a LabSTER simulation next week and keep going, and then eventually we'll hit um, a non-in-class, a non-LabSTER based lab, and we'll talk about that when we get there. All right. So if no one has any questions, we got some slides to get through. Got some slides. As always, if you have any questions, feel free to, along the way, to raise your hand. Feel free to type in the chat, and feel free to speak up. I don't think anyone's ever asked me a question yet, so we're five classes deep. I feel like I'm due for a question at some point. All right, so to genetic code, we're going to start off by looking at this guy, the double helix, one of the most important discoveries of the 20th century, arguably one of the most important discoveries of all time. Because as we'll talk about today, and as we'll keep talking about for the rest of the semester, this guy is really the start and end of everything we're going to talk about. When you talk about evolution, have to talk about this guy. If you're talk about biochemistry, we got to talk about this guy. If we're going to talk about proteins, we got to talk about this guy. Anything in biology, it starts with double helix, it starts with DNA. So we're going to spend a lot of time this semester talking about DNA, but today we're going to start with the basics, what we know, how DNA structure was discovered, what are the rules around DNA, how does DNA replicate it, how do we read DNA, and all those fun stuff. So as you can see, it's going to be very action-packed, as obviously all of our classes are very action-packed with cool science. But we're going to start off actually with an experiment that occurred in the late 1920s by a gentleman who is like myself, was a microbiologist. And this gentleman's name was Frederick Griffith. And what he was doing, he was working on a bacteria called Streptococcus pneumoniae. Now, just as a note, you can tell by its name, pneumoniae, it causes pneumonia. So this is one of the many types of bacteria that causes pneumonia. But you also notice by its name, strep. So it's Streptococcus. If you've ever had strep throat, you can thank this genus of bacteria, Streptococcus, because they cause strep throat. And if you're anything like me, I had strep throat, I think 23 times in 1 year. And so you hate Streptococcus. like really hate Streptococcus. Now, strep pneumoniae has two strains. It has the S strain for smooth and the R strain for rough. The S strain is what we call virulent, and virulent means it can cause disease. The R strain is non-virulent, meaning it cannot cause disease. Pretty straightforward. Now, Griffith, what he did is he injected mice with different strains of these, hoping to understand the difference between these strains. And so what he did is he took the egg, which is living, and he injected it into a mouse, and he saw that the mouse died of pneumonia. And that's pretty straightforward, right? You get pneumonia, you are sort of likely to die, especially if you're a mouse not getting any antibiotics. Next thing he did, he took the non-virulent strain, injected it into the mouse, and the mouse lived. Pretty straightforward, right? Pathogenic bacteria kills, non-pathogenic bacteria does not kill. But the cool part is what he did next. Now, what he did is he took the S strain, which remember is the smooth or virulent strain. He heat killed it, so he just put it under fire to kill it, and he injected that into a healthy mouse, and he found that the mouse did not in fact die. So that makes sense. You have a pathogenic bacteria, you treat it with heat, you kill it, and you infect it into infected into an organism, and it does not cause a disease, thus does not kill the mouse. Pretty straightforward, right? Now, the cool thing here is what he did next. He took the heat-killed virulent strain, so that S strain, and he took the live non-virulent strain, R, mixed them together, and injected them into a healthy mouse. And surprisingly, what he discovered was that the mouse died. And when he re-isolated the bacteria from the lungs, so the mouse ended up dying of pneumonia, when he re-isolated the bacteria from the lungs, he found he could only isolate the S strain, the smooth, virulent strain of the bacteria. So what's going on? What's going on with this, right? He has a non-virulent strain, he mixes it with a heat-killed virulent strain, and all they can isolate out the back end is a virulent strain. So something fun going on here. And what Griffith unknowingly discovered was the process of transformation. And transformation is a pretty cool process. If you're interested in the medical field, I recommend you look up transformation because it's pretty important for how antibiotic resistance is spread within a clinical setting. And if you ever take microbiology here at Fisher, you'll learn all about that. But the cool thing about transformation in Griffith's case is the bacteria that was non-pathogenic, so that R strain was picking up some kind of information from the dead S strain. Right? That's what's going on. The S strain was dead, so it can't cause infection, but the R strain is non-pathogenic. It can't cause infection. So something from that dead S strain was making its way into the R strain. And Griffith had no idea what was going on here. He knew something was being transferred, but he didn't have the terminology or the knowledge to know what was going on with this process of transformation. Now, you should sort of know by the title of this and what I've talked about thus far that what ended up getting transferred here was DNA. The DNA from the deadly strain S was being picked up by the non-pathogenic strain R and changing the R strain into the S strain, making it pathogenic, making it kill the mice. And so this is what Griffith sort of put forth. He didn't know what it was. And we wouldn't know for another 35 years what these bacteria were picking up. But he put forth this idea that some force could transform his non-pathogenic bacteria into a pathogenic bacteria. And that force is obviously has to be DNA. Now, again, he didn't know this, but this is what he put forth. Now, fast forward about 20 years, Avery, MacLeod, and McCarty, they repeated Griffith's experience using purified cell extracts. And they found similar things, but what they actually did to improve on Griffith's experiment is they actually removed all protein from the suspension, and they found out without protein, the transformation process still happened. So what was doing the transformation wasn't protein, it had to be something else within inside the cell. And then what they did is they injected the cells with an enzyme that digested DNA, and it removed the capacity of the bacteria to be transformed. And so this basic experiment, when you think about what MacLeod and colleagues did, and you look at what Griffith did, you put them together with the removal of protein and the removal of DNA, suggests that DNA has to be the causative agent of this transformation. That DNA has to be picked up by the non-pathogenic bacteria from the pathogenic bacteria, thus transforming the pathogenic bacteria into, non-pathogenic bacteria into a pathogenic bacteria. So these are the two basic experiments that put forth the idea of our DNA being the causative agent here of the transfer of information. But again, we still didn't have DNA as a structure or quite understand what was going on. And so we're going to fast forward again 12 more years, and we're going to talk about another really cool experiment by some guys with some fantastic last names, Hershey and Chase. That's a pretty sweet name, Hershey. I don't know. I just think about the chocolate. Now, Hershey and Chase were microbiologists again, and they were investigating bacteriophages. And now, bacteriophage, or a phage, so Phage, are just a class of viruses that only infect bacteria. There are billions of them on this planet. There are, I think it's 10 to the 30 bacteriophages on this planet. There's tons of them. So they're harmless to you and I, but they love to hunt down and kill bacteria. So that's the setting that they were working in. And what they did know from studying these bacteria killing viruses that these phages were comprised only of DNA and protein. And they wanted to assay which of these molecules was the genetic material that was injected into bacteria. Was it protein? Was it DNA? Now, based on our previous experiment that we discussed with Avery and colleagues, right? It doesn't seem to be protein, right? It seems to be DNA, but they still didn't know. And so they did this really sort of fun experiment. And so what they did is they labeled the bacteriophage's DNA with radioactive phosphorus, so phosphorus 32, and they labeled the bacteriophage's protein with radioactive sulfur. Now, if you remember which amino acid contains sulfur, it's cysteine or methionine. There's only 2 amino acids, just as a reminder. And they tracked the radioactive materials. So again, if it was the DNA that was the transforming agent or the infectious agent of their bacteriophage, you would see radioactive labeled phosphorus inside the bacteria. But if it was protein that was essentially the agent of the virus, you would see radioactive sulfur inside the bacteria. What they did find, though, is that the bacteriophage's DNA entered the bacteria, and that was used to essentially infect the bacteria, and you didn't find any trace of the radioactive sulfur inside the bacteria. So what they concluded from the experiment was that DNA was a genetic material, and let me just show you what they did. So they grow the phage in radioactive sulfur, radioactive phosphorus. Now, the idea being is they waited for the phage to infect the bacteria, and the way phages work, as we know now, is that they inject their genetic material, not their protein. But again, back in the 50s, they didn't know what was going on here. And so the idea being is if they found radioactive sulfur, that would indicate protein was the infectious agent. And if they found radioactive phosphorus, that would tell us that DNA was the infectious agent of the virus. And so they did that, and they blended it, they separated, and they found out that in fact it was phosphorus, indicating that DNA was what the viruses were injecting. Viruses were essentially the transformative agent of the virus, and the transformative agent of our bacteria had to be DNA. So these are the three sort of key experiments that led us down the road. Now, you'll notice I stopped at 1952. For those of you that know anything about the history of DNA, the 50s is a really exciting time. We're going to talk about very shortly. Watson and Crick won the Nobel Prize a few years later after this experiment. So what we knew from these three experiments catapulted Watson and Crick to discover the structure of DNA. But we're going to talk about the sort of the basics of DNA structure before we talk about Watson and Crick again. Now, as we talked about in the macromolecules lecture, DNA is a nucleic acid. So it is comprised of nucleotides, and these nucleotides are comprised of a 5-carbon sugar, and this sugar is called deoxyribose, and that is connected to a phosphate group, and this phosphate group is connected to the 5' carbon of the sugar, and I'll show you what that means in just a moment. And then those backbone and the backbone and the sugar are connected to a nitrogenous base, which is either adenine, thymine, cytosine, or guanine. And then there's also a free hydroxyl group, an OH group, attached to the 3' carbon of the sugar. So let's look at what that means. So we have the nitrogenous base here, right? So in this case, we have our nitrogenous base of guanine, right? So this is, you have the table, adenine, guanine, cytosine, and thymine. So these are our 4 DNA bases. And you can see the basic structure, right? The phosphate group here, PO4, that is covalently bonded to the 5' carbon here, so this carbon here, of this 5 carbon sugar. And you'll notice that there is a hydroxyl group at the 3' sugar here. And then the final thing you'll notice is that we have this sort of green box. And you might ask yourself, well, what's that green box for? Well, as you can see, in RNA, there's a hydroxyl group, so an OH group here. In DNA, there's just a hydrogen there. So this is our nucleic acids. This is the basic structure. Know it, love it. And again, this is something that's really sort of fundamental to understanding. Everything you're going to learn from here on out is based upon this basic structure of DNA. So in terms of DNA, in the land of DNA, we have 2 classes of nitrogenous bases. We have the purines, which are adenine and guanine, and then we have the pyrimidines, which are cytosine and thymine. And so you'll notice they have very different structures, right? Our pyrimidines look pretty similar, right? They have the single ring of carbons and as well as nitrogens, but our cytosine differs from our thymine in the presence of an oxygen or an amino group, so the amino group being NH2. And then finally, we have our purines, which is adenine and guanine, which is our double rings here, and they don't differ very much, but you'll notice we have that amino group here again, and the oxygen here, and then you also know we have an amino group here and a hydrogen here, and we're lacking that here. So chemically speaking, they're not all that different. And in fact, to synthesize them, it's a similar set of genes that synthesize all of them, just as a note. And the other thing I'll mention before we go on, DNA has a counterpart. So DNA can be copied into another type of nucleic acid called RNA. And RNA is essentially the same as DNA. Now, the big difference between RNA and DNA here, as we've mentioned, is that the sugar here in RNA it has an OH, and DNA it has an H. And so that's the only basic part here of the backbone, is the difference between an OH and an H. And the nitrogenous bases are the same. However, in RNA, we substitute uracil for thymine. So RNA is A, G, C, and U. DNA is A, G, C, and T, just as a note. So those are our building blocks of DNA and RNA. Very, very simple. But these are our monomers, and we string them together to form polymers, and those polymers are going to be our DNA sequences. Pretty straightforward. These 4 bases make up everything that is new, and that's what I think that's really remarkable about DNA is it's 4 bases. I mean, protein's got 20 amino acids, but DNA has 4 bases, and that's it. And depending on how you string those 4 bases together, you can create some amazing things. Think about everything about you: your capacity to walk, your capacity to talk, your hair color, eye color, right? Your athletic ability, right? That's all dictated by 4 bases strung together in a slightly different order. And I think that is truly a remarkable thing about DNA and how simple it is, but how just by changing little bits can induce complex changes in physical characteristics. I don't know. To me, that's always amazing, and maybe it's not, sort of as amazing to you guys, but I always find that just to be absolutely amazing about DNA. Simple at its basic level, complex at its most sort of expanded level. So, so those are our bases. But to get our DNA together, we have to string them together, and we have to link nucleic acid to nucleic acid, and to right, DNA base to DNA base. And the way we link 2 DNA bases together is through a special type of bond, which is called a phosphodiester bond. And what happens is we form this between the phosphate group of one nucleotide and the 3' OH of the next nucleotide. Just to remind you, so the 3' OH is here and the phosphate group is here. So we're joining this to this between amino acids, between DNA bases. So what this looks like is we link the sugar to the phosphate. And again, you're seeing that COH here, and we're forming the COH would be right here, and we're connecting it to the phosphate of the other group. And that's how we join our nucleotides together. Now, there is an important thing, how they're linked together. Okay, it's got a bond name, that's fine. But the important thing here is that there's directionality. And that's an important part. There is directionality here. So you'll notice I have 5', so 5 with a little, comma, not a comma, apostrophe. And then we have a 3 down here with a little apostrophe. So this is 5' and 3'. So DNA has directionality. It has a 5' to a 3' directionality. So like proteins had an N term, C terminus, DNA has a 5' and a 3'. And that's how DNA is ultimately organized. And the 5' always ends at a phosphate group, and the 3' always ends in the hydroxyl group, just as a note. And just to tell you where they come from, 5' comes from here, and 3' comes from here, just as a note. So that's how we string our DNA bases together. But DNA is a double helix, right? It's not just one linear strand. It's 2 strands parallel to one another and connected. And so to determine how our DNA is connected, we need to understand Chargaff's rules. And so these came along in the 1950s as well. And so Erwin Chargaff, determined through several different types of experiments that the amount of adenine always equaled the amount of thymine in DNA, and that cytosine always equaled the amount of guanine in DNA. And so what it tells us, there's always equal proportions of purines, A and G, and pyrimidines, C and T. So the amount of A and T in a molecule of DNA are always identical. So if I have 50 T's, I have 50 A's. If I have 50 G's, I have 50 C's. So there's always in proportion to one another. That's an important thing to remember. A with T, G with C. That'll again, if you remember that, you'll be pretty good overall. So we have how the base pairing works. We have the A, T, G, and C, but we don't have that 3D structure, right? We don't have the double helix. Well, to get the double helix, we had to actually bring in a really, I guess, I would argue, a pretty badass woman named Rosalind Franklin. Now, Rosalind Franklin, she's a really interesting person. If you've never heard of Rosalind Franklin, I should suggest you take like 15 minutes to read up on her after class. She's a really fascinating woman, and her story is really a powerful one. As not just a scientist, but as a woman in science, and frankly, just as a minority in science. Women are still minorities in science. So she has a really fascinating story. And like I said, if you have 15 minutes or so, please take a read of her story, because she's a really amazing woman. But in terms of why we're talking about Rosalind Franklin today is she used a technique called X-ray diffraction to study the 3D structure of DNA. So what she would do is she would extract DNA from a cell, bacteria, eukaryote, it didn't matter, and she would blast it with X-rays. And as she was blasting it with X-rays, she would look at, she would get these pictures as we see here, this X. And this, from looking at this X, she determined that DNA had to be helical. So think of like a corkscrew. That's what helical means, right? Or you think about like the alpha helices in our protein structure, right? That's what she discovered. Using the X-ray, she found that DNA was helical. So that's another important piece of DNA structure. Um, and then another scientist came along. His name was Maurice Wilkins. That she worked with him as well, and they discovered that the molecule has a diameter of 2 nanometers and make complete turn of the helix every 3.5 nanometers. And that's an important thing. That distance, that 2 nanometer diameter and 3.4 nanometer turn, is conserved across DNA across every species on this planet. So that structure is that she elucidated was really important. So Rosalind Franklin, badass woman, underappreciated for what she did. Read about her. She's awesome. Now, now we're at 1953, so we we haven't gone too far, right? We started at Chargaff was in 1952, Franklin was in 1952, Hershey and Chase were 1952, right? Fast forward to 1953, so making a lot of progress in a very short time here, to the two gentlemen who won the Nobel Prize for discovering the structure of DNA. This is James Watson and Francis Crick. Now, their ultimate contribution and why they won the Nobel Prize is that used all the data that was before them to deduce that the structure of DNA was a double helix. And it's important to note that they won the Nobel Prize from this, but they won the Nobel Prize by using data that other people have. And so they actually didn't do a single experiment related to DNA themselves, but credit for discovering the structure of DNA. And that seems kind of weird. And you should be like, well, why did they get that? And the reality is that Rosalind Franklin and Maurice Wilkins and Chargaff also probably should have won the Nobel Prize with them, in particular Rosalind Franklin, but they kind of got shafted. So just as a note. And I think that's kind of a weird thing. But that's ultimately 1953. We know the structure of DNA from Watson and Crick. Again, Watson and Crick building upon the work of others. I would one thing though about these two guys, even though they get the credit, James Watson's kind of a jackass. And as a scientist, if you're gonna, you know, um, look up to someone as a researcher, as a person, don't look up to James Watson. He's a jerk. He's anti-Semitic. He doesn't deserve the credit he's gotten. Francis Crick, on the other hand, really great scientist, not anti-Semitic, pretty cool guy. But I figured I'd just make mention because, there's the unfortunate reality is that, the sort of the politics of science are still always there. They were clear in the 1950s, and they're still here today. And you guys are going to encounter this when you leave Fisher. So I figured I'd just make note that it was always there and probably going to be there for a while. But I digress. So in terms of the structure of DNA, when we look at a single strand of DNA, it's the phosphodiester linked backbone. So we have the phosphate sugar linked by the phosphodiester bond, phosphate sugar, so on and so forth. And remember the directionality, 5' to 3'. And so you see the sort of the middle of the DNA. It is all of our DNA bases, A, T, G, and C. One thing I'd also like to note is that there is a net negative charge on the backbone of DNA, just as a note. And so Watson and Crick's big thing was sort of this double helix. And so DNA is not single-stranded, it's double-stranded. And it's not just like a regular ladder. It's a ladder, but you twist it into a helix. And so DNA has a couple sort of anatomy points that we're going to discuss. And so there's minor grooves. So if you sort of look at the way this wraps around itself, there's minor grooves between the backbones here. And these are oh my God, I'm forgetting the distance. It doesn't matter. Then we have major grooves as well. So those are the sort of the two big parts. So it's just repeating minor group, major group, minor group, major group, and so on and so forth. That's DNA's repeating structure. It is always between a minor groove and a major group, it's always 3.4 nanometers, and the distance horizontally of DNA is always 2 nanometers. And just as a note, the distance between every DNA base, so every rung of the ladder is 0.34 nanometers. So we're talking really, really small distances here, right? 1 nanometer is, remember, a nanometer is 1 billionth of a meter. So it's very, very small distances we're talking about here. We're talking at them almost at the molecular level here. And the other thing I want to note is that DNA is anti-parallel. And what does that mean? Well, you'll notice here that we have 5' that runs this way, and we have, and then on the other side we have 3'. So we have 5' to 3', 5' to 3'. So they're anti-parallel to one another. And so as we've talked about, DNA base pairing is complementary. A bonds with T, G bonds with C. Now, historically speaking, what we used to think is that A bonded with T and G bonded with C primarily through hydrogen bonds. So this sort of weak bond between oxygens and hydrogens, and nitrogens and hydrogens. So this is how we used to think. We now actually, water plays a big role in holding DNA together, but there definitely is still hydrogen bonding here. It just doesn't seem to be as important as the slide is making it out to believe. Now, every textbook you read from now on will tell you hydrogen bonding and it's how it's told together, but I just want to make note that, like, the brand new hot off the press research shows water actually kind of holds this DNA together, and it's less so important for hydrogen bonds. Just wanted to put that out there. I'd be remiss if I didn't give you some hot off the press research on DNA, because it doesn't happen very often. There's not a lot of research that comes out that's sort of fundamental, like what holds DNA together. It's usually like crazy stuff. So just as a note. Now, that's the structure of DNA. That's how base pairing works. That's how it was discovered. Now, let's talk about how we replicate DNA. So a cell in your body has DNA. Doesn't matter which cell it is. It's a blood cell, it's nervous cell, if it's your sperm or egg, it doesn't matter. Everything has DNA. Now, as a person, you're essentially comprised of about 30 trillion cells, and you started off as one cell, and that one cell had all the DNA that you'll ever need in it. But for you to go from one cell to two cell to four, and so on and so forth, to the 30 trillion cell mass that you are now, you had to make a copy of DNA. And so every cell has a copy of your original strand of DNA. And every time you make a new cell, and we'll talk about this in a few classes, but you have to replicate your DNA. And so we're going to talk about DNA, and there has been 3 models, historically speaking, proposed to explain how DNA replicates. So we have the conservative model, which is where both strands of a parental duplex remain intact, and the new DNAs consist of all new molecules. We have the semi-conservative model, which is the daughter strands each consist of one parental strand and one new strand. And then we have the dispersive model, which is the crazy of all the models, where new DNA is dispersed throughout each strand of both daughter molecules after replication. And you might be thinking, well, what the heck does that mean? Well, I'm going from one DNA strand and making it a copy. So if I have a conservative model, I make an exact copy, and I have the 2 resulting strands are, one is completely new DNA, as you say here in gold, I guess, or bronze or copper, and the original strand is here in blue. Semi-conservative is each new strand of DNA is half old and half new, and then the dispersive one, which again is the crazy of all, is that each new strand that we make of that, again, that's a copy of the original, is like a mixture of new, as you can see here. Crazy stuff for this last one. I don't know why anyone ever thought that was a potential model for DNA, but I digress. And so to understand this, we're going to talk about another really important experiment in the late 50s by Melson and Stahl. And what they were doing is try to understand how DNA was replicated. So which of these 3 models was correct? And so what they did is they grew bacterial cells in heavy nitrogen, so an isotope of nitrogen of 15N. Just as a note, regular nitrogen is 14N. After several generations of bacteria, the DNA of these bacteria was denser than the normal DNA, because 15N nitrogen is heavier than 14N nitrogen. And then they switched the media to containing lighter nitrogen, so 14N nitrogen. And then the DNA was extracted from the cells at various time intervals. Sounds like a complicated experiment, but it's actually pretty fantastic. And so what they found, do I have a, here we go, there's my figure. So what they did is they, just to sort of show you the experiment, they have E. coli with its DNA, and they're growing it in heavy nitrogen. And they're in a media that stimulates their growth. And then what they did is they shifted them to be into a 14N nitrogen medium, again, lighter nitrogen. And what they did is they took samples at different time points. And what they did was they were trying to understand where was the DNA essentially, how is it being replicated, right? The idea being is that nitrogen is the real part of DNA, right? It's the nitrogenous base, right? A, T, G, or C. And so by having different weights of nitrogen here, they can figure out where DNA was being incorporated. Was it being incorporated essentially semi-conservative, conservative, or dispersive, right? That's the idea. And they could do that just by looking at these isotopes. And so what they did is they rejected the conservative model. And so the two densities, so again, heavy nitrogen and light nitrogen, was not observed. So after the first round, so that rejected it. The semi-conservative model was ultimately supported because it was consistent with everything they were seeing. And then finally, they rejected the dispersive model because there was inconsistent results for the second round. And all you need to take away from the experiment is basically that the model that makes sense for DNA replication, how DNA goes from one strand to two strand, is this semi-conservative model. And just as a note, the semi-conservative model is this one here in the center, where we take one strand of DNA, we replicate it, and each of the two daughter strands of DNA here are comprised of half new DNA and half old parental DNA. So that's the model that Melson and Stahl put forth, and this is the model that we know to be true today. So DNA replication is going to be our sort of next topic. Again, it's semi-conservative, and it requires three things. So we need something to copy, and the something to copy is a parental DNA molecule. And it says parental, but don't think of it as like your parent's DNA. Think of it as like, I have a cell, and that cell wants to make a copy, and the original DNA of that is the parental DNA. That's what this is referring to here, just so we're not confused. We need something to do the copying. In this case, it's going to be a protein, in particular an enzyme, or several classes of enzymes. And then we need building blocks to make a copy. And these building blocks are going to be more nucleotides, our DNA, our A's, G, and C's. So this is what we need. Now, there's three steps you need to remember for DNA replication. We have initiation, which is the beginning of replication, elongation, where we make new strands of DNA by an enzyme called DNA polymerase, and then we have termination, which is the termination of replication. So these are our three steps. Again, all focus on this idea of taking one strand of DNA and making it into two strands of DNA. So let's talk about the action of DNA polymerase. DNA polymerase is sensitive to directions. So I mentioned before, your DNA is anti-parallel. So we have 5' to 3', but again, the strand that's on the opposite part runs 5' to 3' in the opposite direction. So that's what we mean by anti-parallel. And so because DNA has directionality, the enzymes that act upon DNA also has directionality. And so DNA polymerase, as an enzyme that synthesizes new strands of DNA, has a specific action. So DNA polymerase, very much like your sort of the directionality of your DNA, essentially creates new strands in a 5' to 3' direction. So what you're seeing in this example here is DNA polymerase is essentially synthesizing this new green strand here, and it's moving from 5' to 3'. So it does have a directionality. It moves in a specific direction. So you'll never see DNA polymerase going from 3' to 5'. It always goes 5' to 3'. And that's an important thing to remember. Directionality is important when we're talking about the synthesis of DNA. Now, anytime we see, just as a note, anytime you see anything in biology that ends in ASE, it always refers to an enzyme. So polymerase, lactase, gyrase, helicase, anything that ends in an ASE is always an enzyme. So it's a good little sort of mnemonic to remember. ASE is enzyme. Now remember, all enzymes are proteins, but not all proteins are enzymes. And remember, enzymes are just proteins that catalyze chemical reactions. And building DNA strands, while you might not think it is just simply a chemical reaction. So, but our enzyme, DNA polymerase, it matches all existing DNA bases in a template with a complementary nucleotide. And so, and our DNA polymerases, whether it's a bacteria's, whether it's yours, whether it's a monkey's or a gorilla's, doesn't matter. They all have several common features. And so they add new bases to the 3' end of the existing DNA strand, which means that the DNA polymerase is moving in a 5' to 3' direction. So again, if we have our parental strand here in green, the DNA polymerase binds 3' end of that parental DNA strand and then moves towards the 5' strand, 5' end of the parental strand. But in terms of the new strain that it's making, again, it's making it in the 5' to 3' direction. It's an important thing to sort of remember about directionality of DNA. Now, and the other final thing that every DNA polymerase needs is what we call a primer. And this primer is actually comprised of RNA. And so you depiction down here, the yellow part here is actually the primer. And now we'll talk about more about the primer, what that means in a moment, but just know that the primer basically tells the DNA polymerase, hey, come over here. This is where we're going to start replicating this brand new piece of DNA. So let's start off with the most simple type of replication. We're talk about prokaryotes, and so these are bacteria and archaea. But in particular, we're going to talk about our friend E. coli. So E. coli is a pretty common model organism we use to understand bacterial physiology. And so prokaryote replication is much more simple, simply due to the fact that prokaryotes have a much simpler genome. They have one small singular circular chromosome. They don't have multiple chromosomes. Their genomes are very small. They're only a few million bases, whereas your DNA is a few billion bases, so much, much bigger in size. And so when we're talking about prokaryotic replication, replication begins at what we call the origin of replication, or the ORI. And it does proceed in both directions around the circular chromosome, and they meet at essentially the termination point. And ultimately the overall structure here is what we call a replicon, and we'll talk about what that means very shortly. Now, DNA is bidirectional, so we have the origin here of our bacteria. So this is origin of replication. So this is where DNA polymerase starts to do its thing. And what we end up having, then we have I mixed those up. We have the origin here. This is where DNA replication starts. And then we have the terminator, or the termination point, here in red. And so what we do is we form two large protein complexes. So these would be like a quaternary structure of protein, just as an FYI, that are called replisomes. And we form these two replisomes at the origin of replication. And one goes essentially counterclockwise, and the other goes clockwise. And what they do is they sort of wrap around this strand of DNA till they hit the termination sequence, and then they essentially split off from one another, essentially giving us this sort of half, sort of that semi-conservative, where we have original strand in blue and new strand in red, and then original strand in blue and new strand in red. So this is what replication looks like. But on a bacteria, it is bidirectional. It starts from a unique origin and ends at the termination point. So nothing crazy complex, but this is what bidirectional replication of our bacteria look like. Now, E. coli, like many bacteria, has 3 DNA polymerases. Now, DNA polymerase 1, or Pol1, acts on a lagging strand to remove primers and replaces them with DNA. So we'll get to what that means in a moment. We have DNA polymerase 2, or Pol2. It's involved in the DNA repair process. So if DNA gets damaged, and we'll talk about that later in the class today, but it repairs faulty or damaged DNA. And then finally, we have DNA polymerase 3, or Pol3, which is the main replication enzyme. So this is what's essentially synthesizing. Now, these other two, again, are like, you can think of them as accessory polymerases. This Pol3 is what's actually physically doing this process that we're seeing here. Now, all of these have, all three have that 3' to 5' activity. And I will say that DNA polymerase 1 also has a 5' to 3' activity for removing RNA primers. And we'll get to that RNA primer thing in a moment. But first, before we can actually sort of start replicating our DNA, we actually have to unwind our DNA. And so DNA, as it's twisted, has a lot of tension to it. And so you have to unwind the DNA. You have to release that tension of the DNA, of that helix. And the enzyme that does that are a class of enzymes called helicases. And so they take energy that they get in the form of AP, or adenosine triphosphate, to unwind that DNA. And once that has been unwinded, we have a class of proteins called SSBs, or single-strand binding proteins, that coat the DNA strands to actually physically keep them apart. And then finally, there's a third class of enzymes called toposomerases that essentially prevent supercoiling. Just as a note, and a specific type of that enzyme is an enzyme called DNA gyrase. And this is the one that's directly involved in our DNA replication process. Now, when we're talking about essentially unwinding, we have to start with our friends, the helicases there, because they're unwinding their DNA. Again, splitting that two pieces of DNA apart. Again, that's a lot of this, they're held together, right? And so when you start to unwind them, it causes what we call torsonal strain. I don't expect anyone to remember that, but torsonal strain is just basically the DNA coiling. Think about, oh, maybe you guys are too young for this, but if you ever use like a landline of your phone, it has the cords. Maybe you guys have never used a landline for, I don't know. I don't know what kids do these days, but for those of you that do know, those landlines of your phone, the cords get all tangled up and stuff like that, and that's exactly what happens to your DNA. This analogy was much better 10 years ago when I started teaching about DNA. Not so good now when no one uses landlines anymore, but I digress. But it essentially gets coiled up like a phone line, and this is what we call torsonal strain. But DNA gyrases, what it does is relaxes this torsonal strain, and this torsonal strain can be like multiple sort of loops like this, or a single loop like this. So DNA gyrase is our hero, and he keeps it from essentially winding back up on itself. Now, DNA polymerase, as we've mentioned, has directionality, right? It can only read DNA in a 3' to 5' direction, and it only synthesizes in that 5' to 3' direction. And so this leads to this process of DNA replication being what we call semi-discontiguous, or semi-discontinuous. Now, the idea being is that DNA polymerase has that directionality, right? So it can only go in one direction. But remember, we have two strands of DNA we're trying to copy. So it can only read one DNA strand in the proper way, right? That 5' to 3', and the other one, it couldn't, right? Because it's in the exact opposite condition. And I'll show you what that means in one sec. So what this leads to is this discontinuous replication where we have a leading strand and then also a lagging strand. And the lagging strand the leading strand is just essentially synthesized in one giant go. So you start at the 3' end and you work all the way down to the 5' end in one consistent direction. Whereas the lagging strand, again, it's in a different orientation. So your polymerase has to jump back and forth, creating small fragments of your DNA that you're trying to replicate. These tiny fragments in the lagging strand are what we call Okazaki fragments. So let me just sort of orient you to this idea so that we know what's going on. So the idea with DNA replication here is we start off with our double helix here, we unwind it, and we start to replicate both strands. And so remember, we have this RNA primer that's synthesized by DNA polymerase 2. Nope, 1. DNA polymerase 1. I always get those confused. And this tells the DNA polymerase 3 to hop in there and start synthesizing a new DNA strand. Now, what you'll notice is, again, we have that 5' to 3', that anti-parallel or orientation of our original DNA strand. Now, you can imagine we have this strand on the bottom, which is in 3' to 5' orientation. That's perfect. That's the way that DNA polymerase reads. So we can have an RNA primer bind here. It recruits DNA polymerase 3, and DNA polymerase 3 binds to that primer and synthesizes from 3' to 5', right? And the new strand is producing is in 5' to 3'. And so what that ends up looking like is just one long continuous strand of complementary DNA being synthesized. So this is all our leading strand, and it is synthesized continuously. But the lagging strand is different, right? It's in the opposite orientation, right? It's anti-parallel, right? So it's 5' on this side and 3' on this way. Well, that's not particularly good for DNA polymerase, right? Because remember, DNA polymerase can only read original strand from 3' to 5'. So what ends up happening is the RNA primer binds, and the DNA polymerase binds as well. So DNA polymerase 3 binds, and it synthesizes from this primer at the 3' end to the 5' end. And what it has to do is jump. Essentially, I'm sorry. It binds here. It synthesizes in this direction, and then it has to jump back and synthesize this way, and jump back and synthesize this way. So it has to sort of keep jumping back towards that 3' end. Because remember, the DNA polymerase can only read from 3' to 5' on the original strand of the DNA. That's important. Remember, 3' to 5' on the original strand, which means what it's synthesizing is 5' to 3'. So I know that's kind of confusing to think about. Just remember, original strand DNA polymerase always reads 3' to 5'. And what ends up happening is when we have this lagging strand, it's discontinuous, right? Because we have these jumps, right? The RNA polymerase binds, primer binds, it's synthesized, you jump back, new RNA primer synthesizes, jumps back again. So this is why we call it the lagging strand. It's discontinuous. It's not one long strand being synthesized. And so what we end up getting is a bunch of these little fragments, typically about 1,000 base pairs in length. They can be longer, depends on the species. But it ends up being essentially a bunch of these fragments, which we call Okazaki fragments. So a Japanese gentleman won a Nobel Prize for this, I believe in the 90s, maybe 80s, for discovering these Okazaki fragments. And his last name was obviously Okazaki. And so this is what we're getting. We have a leading strand, which is synthesized in one go, and a lagging strand that's synthesized in pieces. And the pieces that are produced are our Okazaki fragments. And these Okazaki fragments, as we'll learn, have to be stitched back together by one of our DNA polymerases. So synthesis of DNA occurs at the replication fork, and as you can see, this is our replication fork. And the fork just refers to essentially the opening of the DNA from one strand kept together to two strands split apart. And so this is essentially it's just a partial opening. This is what we call our replication fork, that sort of Y. Now we have another enzyme to discuss called DNA primase, and it's an RNA polymerase. So it's not a DNA polymerase, it's an RNA polymerase. And this is the RNA polymerase that makes the RNA primer. And remember that primer that's created by primase, aptly named for what it does, is where it essentially recruits DNA polymerase to bind to the DNA and thus start synthesizing a new strand. Eventually, though, this RNA primer will be removed and replaced with DNA. So you don't have little pieces of RNA in your DNA. It eventually gets replaced with DNA. Now, leading strand synthesis, as I mentioned, it's a single event. So you have one primer, it binds, and DNA polymerase, after it sees that primer, it starts synthesizing the leading strand all in one go. And it's actually kind of cool the way this forms. And so it forms what we call a sliding clamp. And this is what it looks like. It actually just kind of looks like a clamp at the sort of molecular level here. I think it's kind of cool. And it essentially clamps on with the DNA polymerase, and it just essentially holds it on as it glides all the way down the new strand of DNA. Now, that's our leading strand. Again, it's all done in one go, only one RNA primer. However, our lagging strand is not that simple. Our lagging strand synthesis requires several other types of enzymes. So for this lagging strand, remember it is discontinuous, right? We get those Okazaki fragments. So there's a couple enzymes. So we have DNA polymerase 3, which is very much the same as the leading strand, right? That's the polymerase that's synthesizing the new strand. We have the primase again, but the RNA primer, remember, because it's discontinuous, there are multiple RNA primers that have to be synthesized along the entire length of this DNA. We also have DNA polymerase 1, which at the end of synthesis will come along and remove all these RNA primers and replace the RNA with DNA. And then we have DNA ligase, which will essentially stitch together all those Okazaki fragments. So a little bit more complex on the discontiguous strand, but essentially the same overall process, the same class of enzymes that do all this stuff. And I will also mention that termination does occur at specific sites. In bacteria, there's just one site. In eukaryotes, there's multiple sites. And ultimately, what unlinks the two copies from one another is DNA gyrase. So let's just walk through lagging strand synthesis one more time, just so we're all clear. Now we have to read DNA from, in terms of our polymerase, from 3' to 5'. So we have an RNA primer binds, and we have an RNA primer synthesized by DNA polymerase 1. It binds there, and then we have our DNA polymerase 3 binds and then synthesizes the brand new strand here in blue. Then DNA polymerase 1 jumps, synthesizes a new primer. DNA polymerase 3 comes in, synthesizes a brand new strand of DNA. And then that process repeats over and over and over and over again. I'm sorry, I mixed up my enzymes there. I'm so sorry. The DNA polymerase 1 removes the RNA. Primase is what synthesizes the primer. I don't know what I was thinking on that one. So that's the flow we're talking about here. And again, it's much more complex than leading strand synthesis, which is just continuous, but same overall concept. Now, remember, primase is involved with the synthesis of the primer here on the leading strand as well, and DNA polymerase 1 will also be involved with removing of the RNA primer and replacing it with DNA eventually. So similar processes, it's just slightly more complex for lagging strand synthesis. Now, so as we mentioned, we have this sort of molecular machinery for backbone replication of DNA, which is called the replosome. So it would be a quaternary structure of protein, just as a note, and it's a macromolecular assembly of a bunch of different proteins, mostly enzymes, that are involved in this DNA replication part. Now it has two main parts. It has the primosome, which so kind of a fun name to say, which has primase, which again is producing the primers, case, which is unwinding the DNA, and it has several accessory proteins that help along the way. And then we have a complex of 2 DNA polymerase, one for each strand. So let's look at this monster. And this is what we're looking at. It's a pretty big set of things together. And so when I mentioned sort of accessory proteins, the accessory proteins are typically involved in what we call clamps, essentially holding things together. But ultimately, the way this moves back and forth, whether it's leading or lagging, is all connected together. And so I think it's kind of an amazing thing. And so in terms of this whole complex, remember gyrase is unwinding, is keeping the tension there, so it's not curling back in on itself. Helicase is unfolding this DNA, so it's moving in a consistent direction. So in this case, helicase in this whole complex is moving this way. But again, it's all sort of held together in this replosome. And again, this is our replication fork, right? The splitting of the original strand of DNA, but it's all held together in this replosome. And I think it's kind of a crazy sort of structure to look at, but really this is actually what it looks like. I think it kind of looks like octopus, like holding things together, but that's just me wanting to see things and things that are kind of crazy. So, but this is what we're looking at. The replication fork has a replosome, and this is the way DNA synthesis is occurring. And again, it's all held together. So, the lagging strand, even though it goes slightly slower, isn't left behind or anything. That's all done in parallel with one another. And so let's talk about the sort of the steps here. So, so in terms of how we do this, so synthesis, we have stage 1. So what's happening here is that the DNA polymerase 3, so this is what's synthesizing new strands of DNA, is active on each strand. And primase is essentially synthesizing new primers on the lagging strand. Stage 2, or step 2, whichever you want to call it, the loop itself at the lagging strand, so this is the loop, starts to grow because the leading strand is going slightly faster than the lagging strand. So the lagging strand does have a little extra, slack, which cause a loop to form. But again, remember, we're always going 5' to 3'. But again, the lagging strand can't go in that perfect direction in one continuous go. But ultimately, it's moving in terms of directionality on this figure. It's left to right, or 3' to 5' on the top strand of the original parental DNA. 3. When DNA polymerase 3 on this lagging strand hits the previous bunch, so DNA polymerase 3 is going this direction. Once it hits the previous primer, it detaches, it releases its clamp, and then it attaches. DNA polymerase 1 does remove the primer, but ultimately DNA polymerase 3 essentially will come to this new primer, and then start synthesizing again. And then finally, stage 4, the clamp loader here catches essentially the beta clamp. We haven't really discussed the beta clamp, but just know it holds together DNA polymerase 3. It catches it, transfers the DNA polymerase 3 to the new primer. So it kind of like just grabs it like a hook in you know those games where you have the, what are they called? You have the claw machine, so you try to grab things. It's like that. It grabs onto the DNA polymerase 3, brings it back to the newest primer synthesized, and then allows it to keep doing its thing. And then essentially just keep repeating these processes over and over again. So pretty, lots of steps involved, but it's relatively the sort of the big things to remember here are the things I've been highlighting. Remember that directionality is important. Not so much what the enzymes are called, but what the enzymes are actually doing, right? Relaxing the tension, splitting it apart, right? Synthesizing new strands, putting a primer there, removing the RNA. Those enzymes and what they're doing is what's important. All the text here, it is what it is, but the big picture is what you need to remember from this, not the sort of the specific things. It's mostly the big picture. Now, what we discussed was bacterial replication. Eukaryotic replication is much more complicated. Bacterial replication is easy. They have one chromosome, it's small, right? There's an origin and a termination point. So it's pretty straightforward in terms of what to do. But eukaryotic replication is much more complicated. There's much more DNA, there's multiple chromosomes, there's much more high levels of packaging. So bacterial DNA gets packaged a lot, but your DNA gets packaged a lot. And there's also this overall linear structure to it, which makes it huge. Right? You could, I forget the statistic. You could like stretch your DNA to the moon in one of your cells. It's crazy long. But in terms of how we talk about eukaryotic replication, it's very similar enzymology. So very similar enzymes as bacteria use. They just have slightly different names, but they have very similar activity in terms of what they do. The other thing that eukaryotes do that's a little bit different is that there is multiple replicons. So multiple origins of replication are found on every chromosome. So remember with the bacterial chromosome, we had one origin of replication and one termination point. Well, for every chromosome you have, there are dozens and dozens and dozens of them. So just as a note. And I will also make mention of, in terms of where you rate, it actually varies depending on how old you are, and they are adjusted throughout your life. So when you're early in development, your cells have many more origins of replication to start DNA replication versus when you're old. So like when you're in your 70s, you have much fewer origins of replication on every one of your chromosomes than you do when you're, say, an infant or a ball of cells developing in your womb. Kind of a fun fact there. So, and the other sort of big thing is the eukaryotic replication fork is much more complex. So when the eukaryotes are getting ready to divide in a process called mitosis, and this is a process we'll talk about later in the semester, what ends up happening is those helicases, so those enzymes that are splitting things apart, they bind to the DNA before replication begins, but they don't actually start doing anything. And then later on, we have a subset of them that are activated, and then this replosome itself is assembled. I know that's kind of a weird thing to think about, but just to note, there are differences between how bacteria do it and how we do it. And there is some other things here too. There's different polymerases, there's different priming complexes and things like that. So there are differences between what we do and what our lovely microbial friends do. And I will also make mention in terms of our archaea. So we talked about bacterial replication. Archaeal replication is very similar to actually eukaryotic replication. So the enzymes are very similar, the processes are similar, the way they do it is very similar to eukaryotes, but it is very different, as I mentioned, than from bacteria, which I think is a pretty cool thing, because if you remember back from when we talked about phylogenetics, the closest living relative of eukaryotes in the microscopic world is archaea, not bacteria. So the fact that their enzymes are related in terms of DNA replication is it's a pretty cool evolutionary thing. So that's our replication. That's what we got. There is some other parts of eukaryotic chromosomes that we're going to talk about, and we're going to start off with a particularly cool one. I can't remember which group is doing it, doing anti-aging for their BioWiki. If you're doing anti-aging, you'll find this part particularly cool because we're going to talk about telomeres or telomeres, depends on who you ask, said either way. But these are specialized, essentially, caps of DNA found at the end of eukaryotic chromosomes. And they are specific sequences. They're typically like repeating, like a bunch of T's or a bunch of G's, or it just depends on the species. And so there are these caps. And what they do is they protect the ends of your chromosomes from nucleases or enzymes that break down DNA. And what they ultimately do is maintain the integrity of linear chromosomes. Now, when we're sort of looking at um, DNA replication, these telomeres, or the ends of your chromosomes, is a little bit of a problem. So as we're synthesizing, what ends up happening is we remove a primer from the very end of your DNA. But what ends up happening is that primer never gets replaced. So if at the very end of your DNA, so where your telomeres are, the primer can't actually be replaced. You can't replace the RNA with the DNA. You just have to cut the piece of DNA off. And so you can imagine that's kind of a problem, right? I'm cutting off a piece of the end of my DNA every time I replicate. That's not good, right? You're destroying your DNA, right? That's not good, right? Um, and so there is a class of enzymes called telomerases, and these are synthesize new telomere repeat sequences at the end of your chromosomes. And the way telomerase works, so we have this end of the chromosome. We, this piece of DNA that had its RNA primer removed, but you can't physically add DNA back to it. What telomerase does is it comes in. It's an enzyme, as you can tell by its name. And what it does is it synthesizes essentially a new strand of um, of DNA to go along with it. So fun little enzyme. It essentially acts to keep your telomeres long, because remember, your telomeres are protective in nature. But telomeres are this really interesting group of enzymes, and we're going to talk about this right now because I think they're fascinating. And if you find anything what I said about telomerases to be fascinating, please check out the TED Talk I posted to Blackboard about it, because it's really cool group of enzymes. But in terms of how telomerases work, it's pretty developmentally regulated. And well, what does that mean? Well, there's lots of telomerase activity when you're young, and very low telomerase activity once you reach an adulthood, which is kind of cool. So when you're young, it doesn't matter if you're a plant, it doesn't matter if you're a human, your telomerases are very active. When you're old, your telomerase is essentially shut down. They don't actually get expressed in very old people. Once you hit, like, I think it's if it's 50 in humans, you stop actually expressing these enzymes. So what ends up happening, if you don't have telomerases, your chromosomes start to get shorter and shorter and shorter and shorter. And it's kind of a fascinating thing. As your DNA gets shorter and shorter and shorter, you actually start to exhibit signs of aging. And this is true for us, this is true for mice, this is true for plants. It's kind of a wild thing. So the regulation of telomerase as an enzyme is directly related to how you age. So you can imagine, like, maybe in terms of, to stop aging, maybe we can change telomerase, right? Um, which is pretty remarkable, right? It's like almost like the fountain of youth, right? You could potentially stick telomerases in someone or turn their telomerases back on and potentially stop them from aging. And this is why telomerases are cool. And this is why I suggest, if you think anything I just said is cool, to check out that cool TED Talk. There's a really awesome woman scientist there that talks about all this crazy work she does with telomerases and stuff like that. So they do play a role in aging, but they also play a role in cancer. And so cancer cells actually show an activation of telomerase activity. So cancer cells are, as we'll discuss, are just cells dividing out of, right? That's what makes a tumor. And so because they're dividing so fast, their telomerases get shorter and shorter and shorter faster than a regular cell. So what ends up happening is cancer cells reactivate the telomerases so that they don't essentially kill their own DNA. So really cool roles that telomerases play, both in aging as well as cancer. So I think it's pretty cool. And so the final topic of today's lecture, before we break off into our groups to talk about CRISPR, is DNA damage. And now DNA is really important, right? It encodes everything about you, but because our cells aren't, encased in, impenetrable fortresses of that, that can't let bad things in, DNA is bound to get damaged, and it happens a lot. Your DNA get damaged all the time, like a lot. You go out in the sun and you get a tan, you damage your DNA. You drink alcohol, you damage you do anything, you damage your DNA. So there's lots of ways your DNA can be damaged, which means there's lots of ways that your body and your cells have devised to fix that damage. And so in terms of damage, there's two main types. We have damages to your DNA due to replication. And so DNA polymerases accidentally add the wrong base. So pretend you needed a T, they accidentally add a G. That's the type of error and damage that we can see from our DNA polymerases. That being said, many polymerases have proofreading. So it means they can read what they've done and say, oh, there's supposed to be a T here, and they put a T in there instead of the G that they put in or whatever they put in. So there is proofreading. So that's one way your cells can sort of fix the errors. But we can also have what are called mutagens. And these is any agent. It could be light, it could be chemicals, it can be proteins, it could be any number of different things that change your DNA or mutate your DNA. Mutation and DNA and changing your DNA are the same thing, right? That's all mutation is just change. And this can be through radiation, chemicals, like I said, any number of different things. And so there are many, many ways by which your cells can fix your DNA. And there are two broad categories of this DNA repair. They're specific and non-specific. And we're going to talk about both of those to wrap up this part of the lecture. So the first one we're going to talk about is actually mismatch repair. And what this mismatch repair is like, I have a T, and instead of being bound to an A, it's bound to a G. And so what a mismatch repair system does, it removes incorrect bases that have been incorporated during DNA replication. And it just does it by replacing them with the correct base. It's pretty straightforward, right? Now, to do this, they have to distinguish the template strand and the newly synthesized strand. Because remember, the template strand or the parental strand is the correct sequence of DNA. What's being synthesized is essentially the new thing, which is where the error is going to occur. So they have to be able to distinguish that. In E. coli, what E. coli does is it actually methylates in every A it sees in the sequence of GATC. And what I mean by methylate, you just stick on a CH3 group. That's what methylation means, just as an FYI. So they stick on this, and this is how they recognize between old strand and new strand. And so that's one way. Another common way we have is called photo repair. And so this is a very specific type of repairing. And so we have these what we call thymine dimers form. So this happens when you go tanning. So what ends up happening, or if you go hiking, it doesn't matter. Anytime you go outside and you experience UV light, what ends up happening is you form these dimers. And what ends up happening is you have AA, there should be a TT. But what ends up happening because of UV light, essentially it causes the T's to break from the A's and bind to one another, forming that dimer, which I think is pretty straightforward to understand. But we have this class of enzyme called a photolysase. Sorry, it doesn't look like it's a tough one to say, but it is. I promise. But what it does is it actually absorbs light and uses the energy from light to essentially break the thymine dimer and return the DNA back to its original structure. So a specific set of enzymes for doing a very specific process. And then sort of the final thing is excision repair. It's non-specific. Essentially, we have a damaged region of DNA is removed and replaced by DNA synthesis. So this involves three processes: recognition of damage, removal of the damage, and then the resynthesis. So what this looks like is we have a damage or an incorrect base. We have this excision repair enzymes. It binds to the damage, it cuts it out, and then it recruits DNA polymerase 2, if I'm not mistaken, to synthesize a brand new piece of the DNA. It's all based upon that original strand, because generally speaking, the damage typically occurs in the newly synthesized strand, but it can occur in the sort of the old parental strand as well. So just as a note. So your DNA is not infallible. It can be damaged, and there's a number of ways that your body can, and I say your body, I mean, you should say your cells can physically fix that damage to your DNA. But ultimately the keys for today's lecture is to remember sort of the three sort of the main plots, the not so much the the specifics, but remembering what's going on in the sort of the big function of DNA, right? You have that initiation, you have the the replication, and you have the termination, right? And what's sort of happening along the way in those three steps is what's really important for understanding this. And the other sort of important thing to remember is that directionality of the DNA, right? 5 prime to 3 prime, and how the DNA polymerase can read it, and why we have a lagging strand and why we have a leading strand when we synthesize new DNA. So, so that's going to be the end of our lecture for today. So I'm going to stop recording.